Cleaning validation in the pharmaceutical industry. This is the topic for the video. Cleaning validation is very much required, and the cleaning validation methods are required to be developed. Nowadays, the regulatory agencies are very much stringent on the cleaning validation. See, the pharmaceutical industries manufacture many of the products. The same manufacturing area is used for manufacturing of the different formulations. So it becomes very important whenever the new product or new batch is being manufactured, the area and the equipments are required to be cleaned. If these are not cleaned properly, then there are chances of cross contamination. The contamination may lead to impurities, may lead to failure of the batch, may lead to question on the safety and effectiveness of the formulation. Cleaning validation is a critical operation, critical process in the pharmaceutical industry. And cleaning validation ensures that equipment used in the manufacturing of the drugs is free from contaminants, residues, and microbial impurities. The documented process or evidence that demonstrate the effectiveness of cleaning procedures in preventing cross contamination and ensuring the product quality, safety, and efficacy. So, by definition, the validation is establishing a documented evidence that the system is capable of the performance as it is claiming. That means, if you are doing the cleaning validation, that means you are documenting and providing evidence that the cleaning process is capable of removing all the contaminants, residues, and other impurities or other materials which may lead to contamination. Key objectives of the cleaning validation are residue removal, compliance to the regulatory requirements, and product safety and consistency. Residue removal ensures that residues of active ingredients, excipients, also the cleaning agents and microbial contaminants are reduced to the acceptable levels. Compliance is the objective of cleaning validation, and it provides information regarding meeting the regulatory requirements and adhere to the GMP practices. Product safety prevents cross contamination between batches of different products. Suppose you are manufacturing a batch of diclofenac sodium, and after that you are manufacturing a antihypertensive formulation, for example telmisartan. So the diclofenac batch residue should not go into the telmisartan product. This is just for your easy understanding and example I am giving. So whenever the product is manufactured, different products are manufactured, there should be a evidence that the cleaning method or cleaning procedure can reduce the contaminant to the acceptable level. Then consistency. Validate that cleaning procedures are consistently effective over time. Cleaning procedures should remove all the contaminants, all the previous batch active materials, inactive materials, and also the residues of the cleaning agents. Steps in the cleaning validation. The cleaning validation method is developed first, then the identification of critical areas are done, then the acceptance criteria is established, sampling methods are required to be finalized like direct surface sampling, that is swab, or the rinse sampling, then analytical testing, documentation, then revalidation if required. So these are the steps in the cleaning validation. Develop a cleaning procedure. Define detailed cleaning method including cleaning agents, equipment assembly, and cleaning parameters. How you are going to clean the equipments, that is time, temperature, concentration, and all the uh, all the parameters and method in very much detail are required to be developed and written down. Identify critical areas. So focus on the hard to clean surface areas and the product contact surfaces. See, the products are manufactured in some equipments. For example, you take a tablet formulation. If it is a wet granulation process, so it will use shifter for shifting. It will use a container for holding the materials. Then it will use rapid mixer granulator, RMG or planetary mixer or top spray granulator FBP. It will use milling equipment, blending equipment, compression equipment. If it is coated tablet, then coating equipment. So all these equipments are coming into a train of uh, equipments which are used for the batch manufacturing. So critical areas are required to be identified. Establish the acceptance criteria. So in this, limits are required to be set for residue levels based on the toxicity, solubility, and the therapeutic dose of the product. Sampling methods, that is swab analysis. You might have heard about the swab analysis. Direct surface sampling, that is swab, it collects residues from equipment surfaces. Rinse sampling, analyze rinse water for residues. Analytical testing, use the validated analytical methods like HPLC, TOC, microbial testing to detect and quantify the residues. Generally, the HPLC UV methods are used for testing of the rinse sample or swab samples. Documentation, record all the procedure results and deviations in the validation report. Revalidation, periodically revalidate the cleaning procedure, especially when there are changes in the equipment, products, or cleaning processes. Once you finalize the cleaning process, and if you are changing something, then revalidation is required to be performed. So these are the generalized steps in the cleaning validation. Coming to the challenges in the cleaning validation, first is residue detection, then equipment complexity, regulatory scrutiny. These are some of the main challenges. Residue detection involves a challenge of sensitivity and specificity of analytical method. Complex equipments, some equipments are difficult to clean. These have difficult to clean areas, and also these equipments are required to be cleaned very thoroughly. That is a challenge. Regulatory scrutiny, ensuring compliance with the evolving regulatory standards. So many of the time there are very much regulatory queries are asked onto the cleaning validation. In conclusion, cleaning validation is essential for maintaining product quality, ensuring patient safety, and meeting regulatory expectations in the pharmaceutical industry. So you might have heard also regarding the MACO values that I will cover in the upcoming videos. MACO value is calculated and it is a part of the cleaning validation process. Many of the times, the worst case consideration is taken for the MACO values, and based on that, the cleaning validation process goes ahead. For now, for this video, you can have the information on basics of cleaning validation in the pharmaceutical industry. Thank you.